Hi everyone, welcome back to Gcse. Doing some more organisation topic today and having a look at the other core practical. So this time we're going to be looking at the effect of pH on enzyme activity. Now putting it out into plain English, that just means we're going to look at how changing pH, so acids and alkali conditions, affect how well an enzyme works, so how fast it's going to break a substance down, for example. So we're going to be looking at the variables, we're going to be looking at the method, the results you're likely to look at, and a graph that goes along with them, and then looking at the hazards and risks, because these four things you'll be expected to be able to do in an exam. For example, plan your own version of this sort of practical. So as always, grab some paper and grab those pens and follow along with me. Okay, so setting things out like we did in the last video on the food test practical, we're going to be putting our hypothesis in the middle. So how does pH affect the digestion of starch by amylase? And from that, we can start to look at our variables. So we've got our independent variable, the thing that we change, and we've got the dependent variable, the thing we're measuring to see if that change had an effect or not. So if we look at our hypothesis, you can usually use that to glean what the variables are. So in this case, the thing that we're going to be changing is the pH that we're using, and the effect that we're going to be observed is whether amylase digests starch or not. So how long it takes to digest that starch. It's going to be a colour change, so you could say time for iodine to not change colour or something like that. If you got it set out like I have, just turn that into a little table and that's somewhere you can put your results. Don't forget to leave room for repeats as well, because it's really important to repeat our investigations so we are better able to come up with an accurate mean. Okay, this time we're going to be addressing the control variables straight off the bat. So control variables is things that we have to keep the same, so we know that the only thing that's having an effect on our enzyme is going to be the pH. So we're going to keep the volumes of all of our liquids the same. We're going to make sure we have it as consistent temperature as we can, so using a water bath, and then we have the same time intervals between taking samples. Okay, that's the setup done. Then let's have a look at what we'd have to do in terms of a method. Some exams will ask for an equipment list. I've kept this one as simple as I can. You're going to need boiling tubes, you need a stirring rod, you're going to need a 1 ml syringe or a small measuring cylinder, you're going to need a 5 ml syringe and another small measuring cylinder. You're going to need access to a water bath, so this could be an electronic one or one that you set up yourself using kettle water or using a Bunsen. And then this needs to be set to optimum temperature for whatever enzyme you're using. So the type of amylase you'd probably be using in school, you're looking for an optimum temperature around 35, 36 degrees, somewhere around human body temperature. You'd need your spotting tile and you'd need some pipettes. You'd also need iodine solution, but I forgot to put that on my list when I was writing this down. This practical's a bit complicated to do in real life because it requires some precise timings and keeping track of time, which I'm not great at. But in terms of when you're doing it in school, just follow the instructions really carefully and you'll be fine. So the first thing you do is you get a fresh clean boiling tube, and you're going to put 10 millilitres of starch solution in there. Use your measuring cylinder. To that you're going to add about 10 or so drops of iodine. That will react with the starch and turn from orange straight into that blue-black colour. And you just give that a stir to make sure all the iodine and all of the starch have had a chance to mix together. Next thing you're going to do in a separate boiling tube, you're going to put 2 millilitres of amylase, so that's our enzyme, and you're going to put 5 millilitres of your pH buffer. Now pH buffer is just a special solution that stays at whatever pH you set it to or you make it at. We want to make sure that our pHs are really precise. If they were to go up and down during the experiment and fluctuate, that could affect our results and introduce a bit of error. So you give those a good stir. Right, next thing you do is you're going to take those two boiling tubes and put them into a water bath at about 35 degrees C, as I've said. You can leave those in for about 5 minutes. I recommend using a thermometer, putting a thermometer in each tube, because that way you know once the thermometer in the tube reaches the same as the thermometer in the water bath, you are good to go. You'll just wait for the temperature of the substances in your boiling tube to match the temperature of the water. But usually if you leave it about 5 minutes, that should be sufficient. Right, then you grab your spotting tile and you're going to put a couple of drops of iodine into each of the dots. This is where we're going to be placing our solutions every 30 seconds to test whether or not the enzyme is still working or not. Once you've done that, this is where things get precise with timings. So the next steps have to be done pretty quickly. So you need to mix the two boiling tubes together, so you can either pour the amylase pH mix in with the starch, or the other way around, doesn't matter. But as soon as you've done that, you need to start a timer, and every 30 seconds you're going to suck up some of the solution and then place 1 drop into one of your spotting tiles. Then you need to replace the solution back into your boiling tube. Okay, once you've started doing your samples every 30 seconds, you should, depending on what pH you're using, start to see a colour change in your iodine. On the screen I've got sort of a variation of what you might see, or what I've seen when I've done it with my classes. If the starch is digested, that means it's going to turn into glucose, and a glucose, of course, doesn't change iodine's colour. So any spots that don't change from orange to black, that tells you that that enzyme has been denatured. It doesn't work anymore. You might get some spots that have some little black dots in. I'd say that they are still reacting, but that enzyme is pretty much on its way out. That enzyme is nearly denatured, or it's not working efficiently. Anything that's sort of a lighter blue or that blue-black colour, those are definite positive results. That starch is still there. Once you've done that and you've got your results for one pH, you're then going to repeat all of those steps for other pHs you're testing. Quite often if you're doing this in school and you're tight on time, you probably do one pH and then you'd pull your results with other students. So that's how you do it. A couple of hazards and risks before we move on to the results then. It's not the most It's definitely not the most exciting experiment in the world, nor is it the most dangerous. So the main things you're going to need to note of in an exam or while you're doing it is that iodine can stain, so you need to wear goggles and be really careful you don't get any clothing, especially cotton, because that's going to stain it black straight away, and it's really hard to get iodine stains out. And then any broken glass, because we're using glass stirring rods and glass boiling tubes, you just need to make sure if anything breaks, you clear breakages with a dustpan and brush. And if anyone gets a cut, you just wash the wound and then go and seek medical attention, be it your school nurse or someone that has first aid training. So we'll have a look at the results table now. This is partially made up, partially taken from the internet, but you'll see the sort of pattern expect to see if you're doing it yourself. So in this one, we're going to use shorter intervals between our pHs because that's slightly more accurate in terms of getting realistic values. We're going to be looking at the time taken to digest starch in seconds. So in other words, that's how long it took for there to be no colour change in your iodine spots anymore. Then if you're doing higher tier, you'd also be expected to calculate the rate of the reaction. So rate, you might have known in chemistry, is how much product is changed or how much product is produced in a certain amount of time or change over time. In this experiment, you can use a slightly different rate equation. You can use 1 divided by time. Time just means how much time you measured. So if we look at pH 5, it took 420 seconds to not change colour anymore. So we do 1 divided by 420, which gives us 2.3 times 10 to the power of 3, or 0.0023. It's always best to write in standard form, not just because it's quicker and easier in my opinion, but also lots of questions you'll be penalised and marked if you do not write in standard form. So if we look at our results then, we can see the smallest numbers are the fastest reactions. It took the shortest amount of time for all that starch to be processed into glucose by the enzyme. But are we sure that's the optimum temperature? A way you can work out optimums, or have the most educated guess you can, is by looking at a graph, everybody's favourite. So if we're looking at this just sketch graph, we're going to put time taken to digest starch on our y-axis, pH on our x-axis, and I'm going to sort of just guesstimate here what the trend would look like. So we've got numbers going down and then it goes slightly up again. As the gradient is going down, that means that the reaction is speeding up. So the higher the line is, the slower it is. So it's slow at the beginning around pH 5, it speeds up. And when you get to around pH 7, it's going really, really fast because it only takes 90 seconds. And then past pH 7, it starts to slow down again. Well, if it's going really fast at 7, that must mean it's the pH it works best at. So we can have a guess using our graph, wherever the sort of dip in our trough is the lowest. If we read down on the x-axis, that's roughly what the optimum is going to be. And you can also use this to estimate the time taken for a pH you haven't tested. So if I take a random pH, I know 5.7 for example, and I find 5.7 on my x-axis, I go up with my ruler until I hit the gradient, until I hit this line we've drawn, and then I'll go read off the y-axis the time it would take. Those sorts of questions have come up in exams before, and it's a skill that you especially need to be able to do if you are doing higher tier. Right, that's the pH experiment done. Thank you so much for getting to the end of the video. Hope I didn't bore you too much. I will leave you as always with the thing you've been waiting for, the bad joke. Today's bad joke is going to be related to the digestive system, seeing as we're talking about enzymes. I just finished reading a book about the digestive system. The ending was a bit crap. See you next time.